this is not a terribly advanced class, but the goal of the class is actually to have some fun with some basic concepts in computational biology, computational bioengineering. And the level of software knowledge that you're going to need is not going to be enormous, although I did have a question before we even get started, which is, how much interest do people have in neural network AI technologies at this point? Is that something that you would like to work on in this class as well as in your other classes? Or is that something because we've been thinking about including what are called surrogates, which is how to train neural networks to replace deterministic models. And so if people find that an interesting topic, that's something that we could spend a couple of weeks on. And in particular, I think these days now that the job market for people with basic computer science and computer engineering skills is a little challenging. Having skills in other areas actually is pretty marketable. Over the years, this is never a very big class, but over the years, people who've taken this class have sometimes used their class projects as essentially as part of their portfolio and their resume. And we've had students go on to pretty nice jobs based on the fact that this is a little bit different from the things they do in other contexts. And so we haven't tried until this year, we haven't really tried using machine learning as a core component of the class. And it's not in the syllabus, but Pedro has been working with some undergrads on some machine learning surrogate problems, and he's pretty good at it. And so between the two of us, we thought it might be fun for you to do that. But of course, that wasn't in the syllabus. So I don't want to make a change to the syllabus before we start without running it by you and finding out if that would be something you'd enjoy or not. Again, the point of this class is really to have a bit of fun. So this is E340, 542. I hope you're intending to be here. The previous class seems to have been on swimming. It was a sports class in this room. I don't know. Didn't know that. And our topic is going to be understanding dynamics of biological systems as represented with networks. And we're going to be using a particular computer representation of that called Antimony, which is a very, very simple modeling language. And we'll come back to why that is in a minute. Depending on where you're coming from for a class like this kind of work tends to be pretty interdisciplinary. It has a little bit of mathematics, a little bit of programming, a little bit of biology, a little bit of physics. And it's okay if you're not an expert in any of those areas, or some of them are more familiar or less familiar. And the goal is to make it not that you're going to be an expert on any one of those, but to help you try to build up your knowledge together in all of these areas simultaneously. And so don't be nervous if there are some of the subtopics that we're going to cover that are not so familiar. It's all right. You're going to have plenty of time to do that. And it's really not designed to be a difficult course. Doesn't mean it's not work, but it's really designed to be fun. And I hope it will be. The other thing I'm going to say is that these will always be live streamed and recorded. And so if I'm out of town or if somebody's sick or something like that, you're welcome to join. I mean, the predominantly will be here in the room, but there may be times when it's online only. And the recordings it may be a little while till they're edited and put up, but the recordings will always be available on YouTube, so that if there's something you want to refer to, you'll have access to that. Basic content of the lecture today, we're going to talk a little bit about networks, qua networks, and what we're talking about. And then I'd like to get our hands dirty and do a little bit of coding, very simple coding. And one of the things that I really like about the particular language that we're going to be using is that you have to learn about 5 pieces of syntax to actually start doing things. Now, how many people I think most people in ISC have some Python experience. Is there anybody who's never used Python in the room? No. Okay. The level of Python you need is not very sophisticated. The more Python you have, of course, the more you can do, but we don't depend on a huge amount of Python here. And so today we're going to first talk a little bit generically about what networks are, and then we're going to ask questions about what we can do with them. And ultimately, these kinds of problems are used in biological modeling, in industrial modeling, and in biomedicine. And they show up in things like pharmacology. If you wanted to work for a drug discovery company, these are techniques that you would use there. If you were doing process engineering, these are techniques that might be relevant there as well. They could also be relevant if you're doing basic biological or bioengineering research. Starting at a very high level, we're surrounded by things we think of as networks. The term networks probably means different things to different people. And there's a whole network science center here at IU. They typically work on networks in a very different way from the way we're going to do it in this class, and I'll talk a little bit about that. But in fact, the concepts and the methods we're going to be developing here are quite general. So biology is full of things that we abstract as networks. There are the chemical and metabolic networks that create the components that belong to cells. There are signaling networks that transfer information within cells and between them. There are gene regulatory networks that turn genes on and off inside of cells. And at higher levels, they're what are called physiologically based pharmaco-kinetic networks, which is basically your bloodstream carrying chemicals within your body that move molecules around at a large scale. And actually, from an industrial point of view, that last one is the one that's probably the most commonly used. Nowadays, if you want to get permission to market a new drug, the FDA will require what's called a PBPK, physiologically based pharmacokinetic network model of the drug to understand when you take a pill, how fast is it absorbed in your body, where does it go, how is it eliminated, and so on. And so we're going to do a little bit less of that, but the technologies we're going to develop would be applicable to that as well. There also are more generic applications of these kinds of networks, what are called population dynamics models, where you look at how cells interact with each other. For example, we might have here stem cells reproduce and differentiated to semi-differentiated cells, which then reproduce and become fully differentiated cells, and some of these die as well. And that could be a non-spatial model of something that's going on in a tissue. And there also are what are called outcome networks or adverse outcome pathways, where the boxes and the transitions are a little bit more, perhaps more qualitative. There are particular things like health state that might be understood. And actually building these kinds of adverse outcome pathways is a big deal in the context of drug discovery and things like toxicology. And so these are all conceptually networks. Basically anything where you have nodes and arrows connecting them is a network. And I thought I might start out by just showing you 2 examples of networks. One of them is one that we're actually working on as a research project at the moment. And this is something we will do in class. A very simple gene regulatory network circuit in which a gene here, gene 1, creates a molecule that shuts off the production of gene 2 And gene 2 creates a molecule which shuts off the production of gene 1. And that's called a toggle. A inhibits B, B inhibits A. It's a little bit like a seesaw. And in that sort of situation, what's going to happen typically is that either gene 1 will be on or gene 2 will be on. You can't have both of them on at the same time. And that winds up being a very common motif in gene regulatory networks. And there are experiments in which you can see how this behaves when you simulate it from outside. It has a simple diagram representation, and we'll talk about how these diagrams are drawn and what these mean. A here inhibits B. This T arrow is an inhibitory arrow. A promotes itself. The lollipop arrow is promotion. B promotes itself and inhibits A. And again, later on, we'll talk about how to turn that into mathematics, but there's a standard way of translating a diagram like that into a rate equation that says the rate of production of A depends on the concentration of A and B. That's the mathematical representation. And we can turn that then into a computational representation, and we'll start even today on how to do that. And then we can plot, for example, this is for a given initial amount of B arbitrarily scaled to 1. We started out with, the amounts are between 0 and 1. We started out with about half the total amount of B, and now we start out with different amounts of A. And if we start out with a small amount of A, we wind up with a lot of B and a little A at the end. If we start out with a lot of A, we wind up with a lot of A and a little bit of B at the end. A and B compete, one of them wins. This is time on the x-axis and the amount of A on the y-axis. So this is a rate equation turned into a time series. There is a little bit of fancy mathematics called a phase diagram that we can draw from that, and we'll learn how to do that later in the class, which explains the whole mathematical structure of this kind of simple circuit, biological circuit. In this case, X really is A and Y is B, and we can point to any initial concentration would be some point on this diagram. The arrows say how that concentration will evolve in time, and the little crossings between these colored lines represent what are called the fixed points, the final states of that circuit. And so we can actually then do essentially a full analysis of this little circuit. And so, again, I just wanted to show you some simple examples of the kinds of things that one might want to do. It'll take, because the things we're going to do today are going to be very basic. And I don't want people to walk away from the class today thinking that it's totally trivial, that what we were going to do. In the end, you can do rather sophisticated things, but we're going to build up slowly to that level of sophistication. Question, go ahead.
When you're doing something like this for biology, where it's like a lot of interacting things,
yeah.
Like, I guess for the eye example, if you like introduce the drug to see if it helped, do you generally, would that be like you change the structure of the diagram, or you add something to the math, or like, I guess I'm just, where does like all of the complexities?
Well, so that's a really fundamental question. And the answer, of course, is, in reality, if you have an eye and you put something in it, something will happen. In a computational model, if you don't include the thing that you're going to manipulate, it's not there. And so you do have to think, in a sense, in advance what the situations that you might want to model are. But in that eye example, for example, 2 billion people a day use Procter & Gamble shampoo. Some fraction of those people get that shampoo in their eye. The surfactants that clean your hair also damage your eye. Not terribly badly, but some. And so a company like Procter & Gamble would like to develop shampoos that cause less damage to the eye. And so they would like then to be able to understand how eye damage occurs and how you recover from it. Now, in that model, complex as it is, that's fundamentally a model of damage and homeostasis. That is that effectively what we're assuming in that model is that you're coming in and essentially cutting out some of the cells in the eye. It's almost as if you came in with a knife and cut the eye. In reality, the response of the eye would depend on the particular kind of chemical that you use to injure it. And that's not included in that particular model. In the case of drug discovery, what you typically would do, and I would have to show you the two examples I showed maybe weren't the best ones. Well, the repressolator one that I have here actually does have a little bit of that. If I come back here, I have at the top, it's labeled inducer 1 and inducer 2. Those are molecular species that actually act directly on that circuit. And so I can say, if I go into a Petri dish, in this case, and I apply the molecule that behaves as inducer 1, what will happen to the output of this circuit? And so in a lot of drug discovery applications, you would actually have detailed models of biochemical signaling, for example. You would know that drug X interacts with molecule Y in this circuit, and then you could predict the large-scale outcome of that small-scale perturbation. And in toxicology studies also, that's how it would work. But you do have to know a fair amount of detail about the biology of the system to be able to simulate what's going to happen when you have those kinds of perturbations. In general, drawing these kinds of diagrams is the hard part. People get tired in my world of hearing me say the biology is the hard part. Actually, once you know what the diagram like this is, turning it into computer version of it's not so hard. Getting the parameter values from experiments is not easy, but there are one of our collaborators on some other projects, Eran Agman, who's at the University of Connecticut, has a model essentially of every biochemical reaction in the E. coli bacteria. He has 19,000, excuse me, I got to get backwards. It has 12,000 equations of this kind, representing all of the different chemical processes in one bacterium. There are 19,000 kinetic parameters in his model, of which all but 50 he's actually measured experimentally. And so he can do things like predict, is this bacterium going to develop antibiotic resistance? And so he's funded at the moment by the military, which is interested in understanding antibiotic resistance development. So it is possible to build very detailed models of this kind and to validate them against experiment. In an introductory class like this, I'm afraid we're not going to get very far in that direction. But I do hope that if we get far enough that you'll have the basics that you could move forward in that direction if that was something that you were interested in. In the pharmaco-kinetic models, everybody has basically the same bloodstream, and the distribution of chemicals through your body is basically the same. The blood flows in the same direction. It goes from the gut to the liver to the kidneys and so on. And so those kind of models tend to have always the same structure. They differ typically. The commercial ones would differ. For example, you'd have a model of a young woman versus an old man, somebody who's diabetic versus somebody who's not diabetic. And so a company that was developing a drug would have to know how would this drug dosage change in individuals who have different backgrounds? How would you adjust the dosing schedule based on that? So in that case, the network structure would be pretty well defined, but the parameter values, how big your liver is, how much fat you have, how fast your blood flows, those would be different between individuals. In some of these cases, we don't actually understand the network structure because nobody's mapped it. And that makes it a much harder problem. So, but that's those are great. Those are great points, and I think it's important both to be realistic about what we know and don't know, and at the same time, it's not a counsel of despair. People use this kind of modeling all the time in applications. I'm rather impressed in the sense that the people that have graduated from my group are being hired by industry a lot lately. For a long time, people thought this kind of modeling was basically for scientific fun, for discovery. But now industry, especially pharma, has decided that it needs people who can do this kind of work. That may or may not be of interest. I don't know what your particular backgrounds and interests are, but we do sit in an engineering department, so a lot of people do want to go into industrial positions later on. And so I'm mentioning that as a possibility. And I hope we'll have a chance to come back to that. In some versions of some years, I've spent more time in this first lecture really going over background and these high level concepts. This year, I thought it was more fun if people actually get to do a little bit of coding, at least on the first day. So it's not just conversation. But I'm definitely interested in talking about that more. One thing about this class is that we'll have some homework assignments, but we don't have an exam. It's a project-based class. And the goal, either people working alone or in teams, small teams, the goal is to develop a project that you can put up and share. And that's an opportunity to dig into a question. If there's a particular biological question or bioengineering question that you're interested in, when you want to spend the semester working on that, provided it fits into this general network concept, and then we're pretty flexible about what you can work on. As I mentioned, to do this kind of work, you need to know a little bit about a lot of things. I often say you need sort of a Wikipedia level knowledge of a lot of things. You need a little more than Wikipedia for some things, but you do need to have a bit of biology, a bit of biochemistry, a bit of biophysics, a little bit of mathematics and statistics. And it's fine if you're more familiar with some of these areas than others. And this is a small group, but when we've had bigger groups, I often try to team people up. So if there's some years we've had people who are wet lab biologists come in who've never done any programming at all, and some people who never had any biology at all, and try to team them up and learn from each other. In the summer, we teach a 2-week summer school online, and we definitely we have 50, 60 people, and we definitely try to team people up so that they can learn from each other that way. I'd say that people sometimes ask, why is this course at engineering? It actually started life at the physics department here many years ago. But science is really about discovering principles, and engineering is about applying them. And this kind of work can have both aspects, but we're going to focus really on application. How do you use these ideas to get something that's meaningful? If you're on the science side, you're trying to understand and explain biological phenomena, predict new things, and very often guide experiments. Modern experimental biology and biochemistry are very, very powerful, but it still costs a lot of money to do an experiment. If you can do computer simulations and reduce the number of experiments that you have to do, guide them, that saves a lot of money. And so there's a lot of interest in trying to use computation as a way of being more efficient. I'll say also that, as you may know, the priorities of our science agencies have changed a lot recently. But if you look at the NIH guidelines now, computational modeling of biological systems is one of their priorities. There's something called what they call novel alternative methodologies, NAMs, and making computation a leg of biomedical discovery is a critical priority area in the United States at this point. So if we're going to design things as engineers, we have to be able to predict what they're going to do. And fundamentally, that could be we want to discover new drugs to treat particular illnesses. We might have drugs that we already have, but want to know how to use them more efficiently. We might want to do tissue engineering. We might want to be able to design new kinds of biology that don't exist in the normal real world. We might want to do biochemical engineering. I've worked a lot over the years with our colleagues up at Purdue in their biomedical engineering department. And they actually, it's the equivalent of this course is a required course for their first year graduate students. And in fact, Hayden, who's on the call, is helping write a textbook for the Purdue people on this topic. It has a little bit different flavor from this, but it's philosophically very similar. Course logistics, we're going to meet once a week in the afternoons. There's going to be the Zoom link and the recordings afterwards. The textbook that I've used for the past few years is Herbert Sauer's Systems Biology, Introduction to Pathway Modeling. I like that textbook because it's very, very simple and practical. It maybe doesn't go into the applications as much. It's more a methods book, although it has some nice examples in it. If you want to go into applications more, there's this nice book by Eberhard Voigt, First Course in Systems Biology. There's a nice online textbook from Caltech that's available for free. There's Uri Alon's textbook on systems biology as well. And Herbert's also written some slightly more advanced textbooks that one can use as well. The other nice thing about Herbert's textbook is that you can download it for $8, which is pretty good. And so I'd encourage you to do that as well. And actually, the one of the two little homework problems I've asked you to do is to read the first two chapters of Herbert's textbook, find some other textbook online or in person that you could compare it to, and just tell me a little bit about how they're different, because this is still a fairly new field and people come to it with very different points of view. In the spring, somebody asked me if you were going to have an introductory bioengineering textbook, what would it be? So I went online to Amazon and said, well, what are the introductory bioengineering textbooks? And there really aren't any, because bioengineering is such a big field that there really isn't an understanding of what the core of what people need to understand is. And so I think if you compare Herbert's textbook with say Uri Alon's or Eberhard's textbooks, you'll find that the methods are pretty much the same, but the philosophy is a little bit different. You can learn from that. Again, depending a little bit on your background and interests. There are going to be more or less weekly small homework assignments. I hear it says there's going to be a midterm. I probably won't do the midterm. Depends a little bit on what you want. Sometimes students actually liked having a little bit of an exam at the middle, a small test. There will be a project. I'm going to ask people to put that up on Colab, and so it's released, and I'll ask people to present that project. Everybody here already knows some Python, so the first extra isn't there. Some years people have decided to do a little video tutorial on their project. That winds up being a lot of work, and sometimes people are in too much of a hurry to do it. So I certainly wouldn't mark people. I don't require it, but if people make that effort, I'll give some extra points. And I do ask that people try to get the homeworks in on time, because it's easy to get behind. And I try not to overload people, but I also know that you have a lot of demands, and so I try to keep people on schedule. We will be using computers, so please do bring your computer to class. We're going to be using the Tellurium software. I recommend running Tellurium in Colab. but depending on your taste, if you have a IDE that you like for Python, Jupyter, Spider, VS Code, you're welcome to run Tellurium in that as well. It may look a little bit different. I will come up in just a minute. I'm going to tell you that things are slightly different when you run in Colab from elsewhere. Online, there is a resource called Biomodels, and that is a repository of hundreds and hundreds and hundreds of validated models of this kind. And so if you want to know, gee, how do I model cancer or cell progression, you can go to that database, search that, and it'll give you validated models of that. And those models are written in not in Antimony, they're written in a related language called System Biology Markup Language, SBML, but they're translators that'll convert SBML to Antimony in one line. So you can import those models and then run them. If you want to read about things, again, Wikipedia has good articles. I say there's a handout. It's actually online on Tellurium in the background. I am asking you for next week to read essentially chapters 1 and 2 of the textbook. Herbert has quite a nice set of online materials to teach you the basics of Tellurium, tellurium.readthedocs.io, and I'll reference that a couple of times. Let me just talk about models generically. And you already alluded to that in your comment. A model consists of a model structure and architecture. What is the network? What connects to what? And what are the nodes? And then a set of parameters that describe what are the values at those nodes and how do those nodes interact with each other. Ultimately, if the model doesn't predict something that you can measure experimentally, it's not really very useful. And so it's always worth asking the question, what is it that you're actually trying to get out of a model? Biology is very, very complicated, so anything that we do computationally is going to be a very great simplification of the real complexity. And so when you start building models, actually figuring out what to leave out is as important as to what to put in. And as you were just alluding to, if you leave out something critical, then you can't ask the questions that are related to that. So I think that's a good point. Networks. Networks, I think we all have seen networks in our lives. A network is a graph. You have nodes, you have links between them that define relationships. Now, there are all sorts of different kinds of networks, transportation networks, biological networks of various kinds. And so what those nodes are and what those lines are will be different in different contexts. Sometimes those lines are bidirectional, sometimes they're unidirectional. In biology, they're usually unidirectional. And a lot of the network people here at IU focus not so much on network dynamics, but on network architecture. We have some network and we want to be able to describe what it looks like, what its shape is. And here, we're not going to be focusing on that shape. We're going to be focusing rather on dynamics and how things connect. But we have this idea of nodes. In this case, they could, for example, be chemical species. And we have, in this case, arrows that have a concept of directionality. So in this case, A, B, and C are doing something to D and E. We haven't said what that is at this point. Networks are almost always abstractions. They're in the real world. There's some kinds of spatial or temporal relationships. A network architecture assumes that you can neglect all of that complexity. That may or may not be reasonable. In biology, neglecting that complexity actually often seems very unreasonable, but somewhat surprisingly, it works pretty well. And so this idea that we can abstract the real biological processes where chemicals are moving from one place to another, signals are moving from one place to another, as these non-spatial networks is actually a pretty harsh abstraction. links, these lines represent the relationship between nodes. And in a dynamic network, those links are always going to represent interactions. And in those interactions are going to change node state in some way. And typically, we're going to represent them by arrows. And you're going to see a couple of different kinds of arrows that we use. Those arrows might represent chemical transformation or transport. A molecular species is moved from one place to another. A chemical breaks down and disappears. A chemical reacts with another chemical species. A chemical regulates the production of another chemical species. And so the particular kind of network that we're going to use is going to depend on what lives at the nodes, what the states at those nodes are, the number of cells of a given type, the concentration of a given chemical, the health state of an individual, and the number and category of the links or arrows between the nodes. In biology in particular, we have also arrows that go from nodes to other arrows, which are called modifiers, and that change the action of that link. And that's something that we're going to see a lot of. It's such a basic concept in biology that people almost forget about it, but it's not something that exists so much in other kinds of network science. Now we're talking about dynamic networks. As I mentioned, a lot of people are interested in the architecture, the topology of the networks. But here in our world, we're going to be talking about how networks change in time because we want to be able to control them. And there's this nice article in SciAm from some years ago, which discusses the difference between dynamics on networks and dynamics of networks. And at some level, these things are a bit fungible with each other. But in dynamics of on networks, we have a fixed network architecture, and the state at each node changes in time. And that's typically what we're going to be focusing on here. In dynamics of networks, the network architecture itself reorganizes. And we're going to be focusing primarily on the one on the left. Now, if you're used to things like neural networks, in principle, you can imagine some all-to-all neural network where every possible connection exists, and you change the weights, and you could say that that's actually changing the architecture of the network. And so, in principle, dynamics of networks and on networks are both specializations of some higher level description. Again, we're going to be focusing on dynamics on networks, and so we're going to be assuming that we know network architecture, and then ask the question, given that network architecture, how does the system evolve? And so we're going to have our nodes. Those nodes are going to have states. Those states are usually going to be numbers. The amount or concentration of a particular chemical. If we were modeling the economy, it could be the number of dollars in a bank account. It could be whether a person has the flu, population of a city, the firing rate of a neuron, differentiation state of a cell. And again, the links represent interactions. If you're interested in examples, economists use network models all the time to represent the economy. Neuroscientists represent neural activity in the brain this way. Epidemiologists, and there used to be a lot of computational epidemiologists here at IU, represent the spread of infectious diseases this way. Ecologists represent populations and animal ecosystems that way. People doing urban planning might represent pollutant flows in the system. We've talked about pharmaco-kinetic models already. And then we get back to the kind of models that we're going to focus on a little bit more in this class with things like gene regulatory networks, signaling networks, and metabolic networks. The key thing in the dynamics on networks is that we're going to have some set of rules, some set of rate laws that say that given the current network state, how does the state of the network change in time? And that kind of mathematical representation, what are called rate equations, are going to be quite universal, and they're used across all of these fields. They're used in economics and all of these other fields in the same way. And so anything that you learn in this class about biology will generalize to these other contexts as well. In our case, we're going to start out with a chemical reaction. Has everyone here had some chemistry course at some point? Has anybody never had a chemistry course? It's fine if you haven't. I want to know, and don't be shy about saying, "Hey, I haven't had this idea. Please stop and slow down and cover it." Because if there's something that I say that's not familiar, it's probably not familiar to other people too. Especially in a department like ISE or in computer engineering, computer science, people come with very different backgrounds. My goal is to try to help everybody get up to speed on all the basics. And so don't be shy about asking for clarification. And so we're going to start out with this basic idea of a chemical reaction. And we'll come back to this in more detail in the future. And people often don't think of a chemical reaction diagram as a network. But if I look at this chemical reaction, here are two molecules of adenosine diphosphate, and they're reacting to form a molecule of adenosine triphosphate and one molecule of adenosine monophosphate. That's called a phosphorylation reaction. Actually, the backwards one is the phosphorylation reaction. So that's the phosphorylation reaction. The backwards one is the dephosphorylation reaction. And you'll notice that there's this little arrow, and it's a bidirectional arrow. I can have two molecules of ADP producing an ATP and an AMP, or backwards. And so this is, in a sense, a very trivial little network. I have a node here, which is ADP, and two nodes on the other side, which are ATP and AMP. And I have a statement that if I use up one molecule of ATP and one molecule of AMP, I can get two ADPs. So that gives me a relationship. If I know what the rate of that conversion is, then I have a set of rate equations. I can have a more complex set of reactions. Two molecules of A become B. B becomes three molecules of C. C and A combine to make a molecule of D. In chemical reactions, mass is conserved. If I use up oxygen on one side, I have to have oxygen on the other. A lot of the biological networks that we're going to represent don't conserve mass, but chemical reactions intrinsically conserve mass. And you're going to find that the software we're going to use, Antimony, was originally designed to write chemical reactions. And so it actually expresses everything in terms of this kind of chemical reaction diagram. Any questions about that so far? We're going to get a little bit, I realize that may be sort of abstract. We're going to try to get things to be more concrete in a minute. So in a chemical reaction, the nodes are going to usually represent molecular species, and their state is going to represent either the amount or concentration of that chemical species. Typically, in chemical reactions, we use concentration because the rate of reaction depends on the concentration. In the case of pharmacokinetics, you actually have to use amounts, so you have to convert those back and forth. Links, in this case, represent either chemical transformation from substrate, which is the left-hand side, the input, to product, the output side. Now, normally, in reality, there's always forward and backward reaction of any chemical reaction. Mathematically, the forward and the backward reactions are considered separately. So you say 2A goes to B at some rate, B goes back to 2A at some rate, and you treat those as separate. Any link, any transformation is associated with some kind of rate of action. You'll discover that in the literature, very often these arrows are just written bare, like this top one. But in fact, there's always some implied rate law that's associated with an arrow. And I will try always to, when I show arrow diagrams, I'll try to always label them with the rate law that we're going to use. If we're going to do more than that, we have to have some idea about what those rate laws might look like. I'm not going to give you a detailed chemistry course at the moment. If you're interested in rate laws, Herbert has a whole textbook on rate laws. But basically, we're starting out with the idea of a chemical reaction. We have some set of molecules that are moving around. To react, they have to find each other and collide with each other. And that gives us actually already some insight into how the rate of reaction depends on concentration. So let's start out with a very, very simple chemical reaction. A plus B goes to C. That is one molecule of A and one molecule of B combined to make one molecule of C. Well, if we want to do any more than that, we want to know how the rate of production of C depends on the amount of A and B. Then we want to be able to make first a mathematical statement of that, and then we want to make a computer representation. Well, the first thing that we're going to do is we're going to focus on the amounts or concentrations. Typically, when we see the regular uppercase letters like A, B, and C here, that represents the names of the chemical species. And we're going to use the italic version of those to represent their concentrations. And we'll typically write the rate or velocity of reaction, here VF, and decorate our arrow. A plus B goes to C at a rate VF. Well, what could VF depend on? Well, it certainly could depend on the temperature in the environment. It could depend on the pressure. It could depend on the pH. But it absolutely has to depend on the amount of A and B that we have. And if we're only looking at the forward reaction, it shouldn't depend on the amount of product that we have. And so we can say that the rate at which A and B make C is a function of the concentration of A and B. Now, there already are some assumptions that we're making there. In reality, chemical reactions are discrete. You have two molecules that come together and react. That's a discrete process. And we'll learn pretty soon how to model those kind of stochastic events as well. But for the moment, we're going to assume that our concentrations are high enough that we can approximate those reactions as a continuum. And there's a whole mathematics. If we were in a chemistry course, we'd have a whole mathematics of when we can make those continuum approximations and when not. The next thing that we have to do is, once we've said that we're going to approximate things as a continuum, now A plus B goes to C at a rate VF A comma B becomes what I was talking about beginning, the rate equation. Every time we have a reaction, we lose a molecule of A. So dA by dt is equal to minus VF A of A and B. dB by dt is minus VF A comma B. And we're creating one molecule of C. dC by dt is VF A comma B. So we've actually now taken that diagram and turned it into some mathematics. But until we know the functional form of VF, we can't do any more than that. Now, the simplest kind of rate law that we can have, and there are many different forms of rate law, is called mass action. And this is based on something which is blindingly obvious, which is that if we don't have any A, we can't have A react with B. So VF of A comma B has to be 0 when A is 0. And similarly, VF of has to be 0 when B is 0. And if we write the simplest mathematical form that satisfies the assumption that VF of 0 comma A, 0 comma B is 0, and VF of A comma 0 is 0, and It's a positive number because we're having a forward reaction. The simplest form we can write down is that VF of A comma B is K times A times B. And that's called the law of mass action. And if you take a chemistry course or a biophysics course, they will actually derive that and show where that comes from. But here we're going to take it as a given. And now we can plug that in here, and our little arrow diagram now turns into an instantiable set of equations. dA by dt is minus K times A, concentration of A times B, and so on. And so now if I give you the rate constant K and the initial amounts of A, B, and C, I can calculate the concentration of A, B, and C at all times. In particular, if A, B, and C start out positive, these are concentrations, so they better be positive. They stay positive in time. It happens that this very simple equation has a couple of properties. If A and B are going to decrease monotonically in time, C will increase monotonically in time, and that will continue until we run out of either A or B or both. And so now we have a very simple mathematical equation. And now we're going to do a little bit of coding to implement that equation on your computer. And so I hope people have their computers, and we can start doing a little problem. So the first thing that I'm going to talk about is using this antimony and tellurium package. And antimony is a language for specifying those kind of arrow diagrams and rates. Tellurium is a Python implementation of that package. And you could say, why do we need two names for the same thing? And the answer is that Herbert Sauer at the University of Washington, who invented this system, loves to give things different names. And so every few years he comes up with another name for his product. They're all a little bit similar. They're always fancier. Each version is fancier. But instead of being antimony version 1, antimony version 2, antimony version 3, every time it's a new, he updates it, he gives it a new name. So you can run Tellurium in Colab, which will be the way that I'm going to do it. And the reason I'm going to do it that way mainly is because it makes it easier for me to share things with you. But again, if you would prefer to run it locally, you can. If you like Jupyter Notebook, you like Spider, VS Code, and so on. Does everybody have if you have a Google account, you automatically have a Colab account. Does everybody have a Google account? Does anybody not have a Google account? No. Okay, fine. How many people have used Colab before? 2. Okay. How many people have used Jupyter Notebook before? A couple of extras. So 2 people who haven't done. Okay. It's not terrible. If you haven't used Colab or Jupyter Notebook, what did you use when you did Python?
I did this.
You used VS Code? What about you? Sorry? Visual Studio also. Okay. You can certainly run these in Visual Studio if you'd rather, or VS Code. Things are going to be slightly different. I have created a template notebook if you're going to be using Colab. And I've created a tiny URL, E340 Colab template. And I'd ask people to open that notebook and create a local copy of it if possible. If you don't have that and you're going to use VS Code, I'll show you about the installation in a second. Tellurium is a Python library. And of course, Colab is a Python notebook system. And so whether you're used to VS Code or Colab or Jupyter Notebook, they're all pretty similar. I'm going to be sharing things primarily in the form of these Colab notebooks. Those will be read-only from your point of view, so you'll have to make a local copy of them if you want to run them and use them. And then there are a couple of issues with Colab. One minor annoyance with Colab is that it tends to put things in its own directory, which may not be the place you want to put it. So, and it doesn't remember what your working directory is. So when you save things, you sometimes have to keep walking your directory structure to put them where you want it. That sense VS Code may be more convenient for you. The other issue with Colab is that it has a few inconveniences. Colab comes with a bunch of preloaded libraries. It assumes that you're going to be using matplotlib and numpy and so on. It really assumes that you're going to be doing things like running PyTorch and TensorFlow, which we probably will be doing a little bit later in the class. However, the particular versions of the one thing that's what Python started out, it was very simple. Now that there are lots and lots and lots of libraries for Python, there are compatibility issues. And so Tellurium assumes and actually loads its own versions of NumPy and so on. And those are not always going to be compatible with the ones that Colab has decided that you're going to use. And so the reason I created this little template notebook is to make sure that you don't have compatibility issues if you're using Colab. So that's why I'd like people to make sure that they use that particular Colab notebook. And maybe, Pedro, if you're there, you could share in the Zoom chat the link for that notebook. If you look at the code, actually, I had a different set of instructions to do the same things. And then, as usual, you ask ChatGPT and it said, oh, it would be better to do it this way. They actually do sort of the same thing. % capture simply hides a lot of the chatter that you get from the installation. Otherwise, you'll get lots and lots of lines installing this, installing this, installing this, installing this. Pip install upgrade pit setup tools wheel is going to give you operating system control. That's the thing that ChatGPT gave us. It's a little bit different from the way we were doing it before. We're then going to install a slightly older version of NumPy. That's what we have to do. Pip install -q Tellurium is the main thing that we need. This pip install diagrams is something that we don't need right at the moment. Pedro was using it, and so we have it there. Import IPython, and then IPython application instance kernel do shutdown resets the kernel and allows you then to load NumPy, matplotlib, and so on. The first time you run this, when it gets to line 7, it will throw an execution crash due to unknown error. Don't worry about that. That's what that line does. And then you can keep executing your notebook as expected. You should only run that install line once. It's going to take a while to run. It'll take about 60 seconds to run the first time you run it. Once it's run once, for the rest of the day, you can use the other cells and it'll be fast. But the installation is a little bit slow. If you're in VS Code or your local Spider or anything like that, then you don't need all of this extra detail. You should be able to just do pip install Tellurium and it should work. So you don't need the extra stuff. If you get version incompatibilities, then you may have to do this locally. It depends a little bit on what you've got installed. If you use a Conda environment, then of course that ring fences things, and then you can tell it which versions of the code to use. If you want to run Tellurium locally, if you go to sourceforge.net projects py Tellurium, you can download a Spider environment which has Tellurium pre-installed. That may or may not cause problems if you have other environments already running. And in that one, you do a pip install in the console, and then you type your code in the left. And it knows quite a bit about how Tellurium works, and so it gives you some nice features. Colab also has a lot of nice features. It has some pretty sophisticated AI debugging and things like that. I certainly don't discourage people from using ChatGPT or Colab's AI or anything else. But what I'll say is this, which is just the way if you use ChatGPT to translate English into French, if you don't speak French, you don't have any idea whether the French is any good or not. And the same thing here, you really do have to do the work to know the basic syntax and understand how the software works to be able to use the AI assistants effectively. And so I'd ask you, by all means, use the AI assistants to help yourself out, but it doesn't get you off the hook from learning the basics of the syntax and the basics of the way the things work. Because I promise you that it will give you things that are 90% correct, but if you don't know the basics, you're not going to be able to fix them. So it helps, but it helps. The more experience you have with the languages, the more the AI systems help you. It would be nicer if they helped more at the beginning, but they help more as you go further on. So if you're going to be using a Conda environment, for example, if you're in VS Code, Conda create -n Tellurium Python 3.9, Conda activate Tellurium, Conda install pip install Tellurium will get that running for you. If you go to https://tinyurl.com E340 Colab template, you should be able to open that. And that you should be able to do. Once you go to Colab, you should be able to find that directly. Does that work for people or not? If you're going to use something like VS Code, then you're going to have to do the install, the pip install, the download, do the pip install. Yeah, so my recommendation is that for today, everybody use Colab, and then if you want to switch to, if you prefer to run local this evening, you can try downloading and installing the things and playing with them. Because depending on your particular configuration, it may be a little bit different. If you go to Herbert's Read the Docs, he has a lot of detail about how to install it on a Mac, how to install it in VS Code, how to install it in Jupyter Notebooks, and so on. But I can't predict beforehand exactly what you'll use. So Colab at least is Colab for everybody. Again, if you want help, you've got this one page. The great thing about Tellurium is it's simple. So there's a one-page handout on Tellurium, which has 90% of what Tellurium is. There's also this great Tellurium readthedocs.io that has manuals, explanations, simple, complex examples. Really, it's an excellent tutorial. And the textbook is written entirely assuming you're using Antimony and Tellurium. So it's also, all of the examples for the textbook are in that language as well. Okay, so the first exercise that we're going to do is going to be modeling a chemical reaction in Tellurium. And again, if you've opened that tinyurl.com Colab template, that would be what we need. And I'd ask you to go online to tellurium.readthedocs.io, click on Quick Start, Simple Example, select the code in that simple example, and paste it into the third cell in your template. Now, if you're doing this in VS Code or something else, it's going to look a little bit different. You're going to have in the bottom right of your computer, you're going to have your console. In the console, you will do a pip install tellurium. And then in your code block, you're going to do import tellurium as TE, and then you'll paste your code into the next cell. Does that make sense for people? If it doesn't make sense, I can make these things bigger. At this point, I'm not expecting you to be typing it from the code that I'm showing here, although I can certainly do that if you like. If you get stuck, I actually created a Colab notebook with the thing typed in it, tinyurl.com, E340 exercise 1-1-1. The only thing that you need to see is to copy the code. I can, if you people want, I can do that here. So I'm going to go to quick start. I'm going to go to simple example. I'm going to copy simple example. I'm going to go to my Colab template, and here where it says placeholder to load antimony model, I'm going to paste that code. Now, if you were running this on VS Code or Spider, the only thing you would need is pip install Tellurium in your console. We're not actually going to use NumPy, matplotlib, but you will need import Tellurium as TE, and then your code. Okay. Let me make this bigger so people can see it. And so now let's see what happens if we try to run it. It's going to take its time here, doing that initial install. Once you've done it once, it's pretty easy. The first time you do it, it's a little bit awkward. In my experience, it takes about 60 seconds to launch. Yep, almost exactly 6 seconds. And you'll get this message, your session crashed. And you can ignore that. And now I can go to my next cell and hit import, and that runs. And now my little simulation is here. I hit run, and it plots. Don't rerun that first install. You shouldn't for a whole day. If at the beginning of class you run that set of installers once, you should be clear for the rest of the day in your cells that are below it. You don't have to run it again. Of course, if Python gronks, then you have to start it again. Again, the install on VS Code is going to should be a lot faster than this. Okay, everybody got that running? Everybody got a plot? Great. Ellen too. Great. Good.
And in VS Code, you install the only one.
Right.
Install every time.
Yeah.
Like the normal package, like matplotlib.
Yeah. Yeah. All right. So everybody should have seen that. So let's actually look at what's in there, and this may be a little bit small. This is going to, and we're going to come back to this next time. This is now talking about the syntax of antimony and tellurium. You have import tellurium as TE. That's the library we're using. And then you'll see a command R equals te.load A. And tellurium can read from files or it can read from a string. And typically our model is just a string of characters. And if we remember in Python, the triple quotes, triple double quotes or triple single quotes allow us to have carriage returns in the middle of a string. So that's why you have this funny triple quote syntax. So R equals TE load A triple quotes. Whatever is between those triple quotes is the model definition in the antimony language. And at the end of it, we have triple quotes, close parenthesis. And then we have a command, result equals R dot simulate 0, 40, 1000. So that is going to run, TE load A is going to load our model and compile it. Result equals R dot simulate is going to execute that compiled model. And again, we'll do that many times. So now we're going to take the chemical reaction A plus B goes to C, and we're going to write it. And we already have now an example which has code that runs, but it's not quite the code that we want. And so we're going to model the code that we already have running to represent the chemical reaction A plus B goes to C. And what I'd like you to do is simply type. The great thing about antimony is that what we type looks like a chemical reaction. The chemical reaction A plus B goes to C is written. Here we've named the reaction R1 colon. That simply says that this is reaction 1. A plus B, arrow C, semicolon, and then the rate, K times A times B. And then we're going to have to give the initial amounts or concentrations. Initial A is 10, initial B is 10, initial C is 10. Maybe I should have made it 0 to begin with. K equals 1, and everything else stays the same. So why don't can people see that well enough to type it? It's not a lot to type. So why don't you try typing that into your code and see what happens. It's not the most exciting chemical reaction in the world, but if you can write this one, you can write one with 100 of the 100 chemical reactions instead of 1. And you can tell that the antimony language is descended from C++, well, from C, because it uses semicolons as separators. And in fact, carriage returns don't do anything in this language, so you could have semicolons between everything and run it in big one line if you wanted to. Unlike Python, antimony does not care about spaces or indentation. It does care about upper and lower case. Let's talk about the basics of antimony and tellurium syntax. There are only really 5 concepts, which are all on the handout. Indentation and spaces don't matter. Capitalization does matter. And it's really easy to type capital K instead of lowercase k or something like that and have a syntax error. I strongly recommend ending all lines with a semicolon. And we're always going to have a mixture of antimony and tellurium. And the one place that really gets confusing is that exponentiation in tellurium uses the caret, and in Python it uses star star. So if you want to raise something, square something, in tellurium, antimony, it's caret 2, and in Python it's star star 2. Okay. So we're going to write the chemical reaction A plus B goes to C, and we type it about as naively as we could. R1 is the name of the reaction, reaction 1, colon, names it, A plus B, arrow C, semicolon, and then the rate. We had that import tellurium as TE already. We have that TE load A triple quote because we're going to have carriage returns. We're allowed to name the model if we wanted to, although we're not going to do that here for later reference. And there are a lot of other things that we could do. You could create compartments. There are all sorts of fancy features, which we're not going to use. So the first thing we have to remember is TE load A loads the model. Anything inside the string gets loaded. Again, the fundamental concept in antimony is a chemical reaction. A plus B goes to C, and the semicolon and the rate. Antimony expects us to define the initial concentrations of A, B, and C, whatever chemical species we have. It will default to 0 if we fail to define the initial values for our chemical species. It is required that any parameter have a number associated with it. If we did not say K equals 1, antimony will crash. You'll get an error. One thing about having a model string of this kind is that when you do a load A and there's a syntax error, the messages you get tend to be fairly uninformative because there are no line numbers. And so if you get a crash when you try to load an antimony model, the first thing to look for is mismatched parentheses. That's the easiest way to have a crash. If you don't have mismatched parentheses, my advice is to start commenting out lines from the bottom and adding things back. And we'll go over the debugging more next time. Okay. Once we have loaded the model, we will say result equals r.simulate, initial time, final time, and the number of intermediate time points we're going to evaluate over. Result will be an array, which will contain all of the output of our simulation. And we can actually control quite a bit that way. R.plot will plot is a convenience function in Tellurium that will plot the output of result, but I could use matplotlib functions to do plotting as well. And that's really all you need. I mean, the coding part of this is pretty simple. There are all sorts of more sophisticated things we could do, but we could actually do an amazing amount with just this. Trying to think if there's anything really lurking in here that's likely to cause you a problem. I don't think there is at this point. Any questions about the syntax or about how you run things at the moment? I'm always torn with this first lecture because some years I jump in and do things that are fairly sophisticated. And some years I think, well, it's better to do things slowly. I promise you things will get more interesting. It's not, despite the fact that the language is simple, you could do a lot with it. So I hope it's not so simple that you get discouraged and say, boy, this is trivial. We want it to be easy, but we want you to be able to do complicated things with easy coding. That's the goal here. And so now we're going to run it. And people know how to run a notebook, so I don't think I have to tell you that. And you should have gotten a plot that looks like that. Did you get something that looks like that? Now you'll notice that most of the thing, for the particular values that I picked, everything is over by a time of about 10. And I plotted for 50 in this case. So I could change my simulate, instead of saying result equals r.simulate 0, 40, try making it result equals r.simulate 0, 10. And see what it does. The other thing that you might want to do is look at what actually that result is. And so result is an output that contains the information from the simulation. And so if you go into a cell and just type result return or print parenthesis result, you'll see the structure here. The first column in result is going to be the time. The second column will be the value of A, the third column will be the value of B, and the fourth column will be the value of C. And this object actually is keyed, and I'll come back to that, so that the columns are named time, A, B, and C. So I can actually do slicing not only by the first column, second column, third column, but I can say, give me the time column or the A column or the B column or the C column. I think in general, working with antimony and tellurium, the most inconvenient things are slicing arrays. Python has all these functions for manipulating arrays, VStack, HStack, and so on. And that's a case where things like the Copilot or ChatGPT are always helpful if you don't remember the exact syntax of how to do a VStack or an HStack. All right. So I'd ask people just to print or display result to see to see what the code looks like. I mean, what the output looks like. Everybody able to view that? Okay, great. So the other thing that I suggested doing was, instead of simulating for such a long time, change your simulation to result equals r dot simulate 0, 10, 100 to zoom in and get a little bit more resolution. You should get something just like that. Well, that's a pretty trivial simulation. So we could change the value of k, the rate at which things happen. That'll stretch our time scale to the left or the right. So you can go back in line 7 here and change k from 1 to 0.1 or 0.01, and you'll see that the time scale will change linearly with that value. Again, today we're going to be doing some very elementary simulations. If you go to the Herbert's, read the docs, he has some rather elaborate examples, and I'm going to ask you to cut and paste 1 or 2 of those examples into your template and run it. And we've already seen at the beginning of the class a little bit some more sophisticated examples that you can get as well. But conceptually, they're not much harder than that. Okay, so here's what happens when we change the value of k. Not that exciting, perhaps. So now let's, we can change the initial value of a and b. I mentioned if we run out of, this reaction will change until we have either a go to 0 or b go to 0. What happens if we start out with a equals 0? Anybody? What's going to happen if we start out with a equals 0? Right, nothing will happen because we don't have any, we can't have the reaction. So we'll just get horizontal lines. That's right. It's always a good idea to test yourself when you do these things, to ask, can I determine what's going to happen? So to make sure that things behave properly. In these very simple cases, it may be obvious, but when we start writing more elaborate models, it's not always clear that if we make a mistake, it's good to have some basic understanding of what we expect. Something else that I haven't really talked about is that antimony is what's called the declarative language. Have people used any other declarative languages? People are, of course, using Python, so you're familiar with object-oriented languages. But declarative languages are a little bit of a strange beast. In a procedural language, if I change the order of my lines, it makes a big difference. If I take Python and I randomize the order in which I put my lines of Python, I'm going to get something that doesn't work. In a declarative language like Antimony, the order of the lines doesn't make any difference, because we're representing that network, we're implementing that network architecture. The order in which we define what piece of the network when doesn't matter, because fundamentally what matters is the network structure. That's something that has some issues. The other thing that happens is that values like a equals 10, that assignment is only done at load on the model. It's only done once. And so we're going to come back to that. There are different kinds of, just the way Python has an assignment equal sign and the double equals for comparison as equals, are 2 things equal to each other. Antimony is going to have 2 kinds of equal sign: the regular equal sign, which is evaluated only once when the model is loaded, and colon equals, which is updated continually as the model runs. And so there are going to be some differences Most of the time, the fact that it's a declarative language isn't going to make a big difference to us, but occasionally it may not behave exactly the way you expect if you haven't used declarative languages. Okay, so yes, so if I start out with no A, nothing happens. And I can change the initial values of A, and I don't want to, since we're going to run out of time, I don't want to take too much time on that one. But what's going to happen is that the reaction will continue until I run out of either A or B. If I start out with more A than B, I'll run until I run out of B. If I start out with more B than A, I'll run until I run out of A, and I can get these different results. So even this very simple trivial this trivial relationship has somewhat different temporal and final outcomes depending on my initial conditions. Okay, so now let's actually do something that's slightly less trivial. And this is actually a little bit more interesting, which is see if you can now take the code that you have here and write what's called a cascade chemical reaction. I'm not going to have A plus B go to C. I'm going to have A go to B, B go to C, C go to D. And I'll start out with 10 units of A, no B, no C, no D. And to begin with, you can make the rate constants Ka, Kb, Kc all 1. And this is actually a somewhat non-trivial result. When the Ks are all equal to each other, it's rather trivial. But if you start playing around with the Ks not Ka, Kb, and Kc not equal to each other, you can actually get some rather interesting results even from this very simple model. As a heads up, if you're going to cut and paste R1 to R2 to lines, it's not allowed to have the name of a chemical reaction reused. If you type R1 colon A goes to B, and then R1 colon B goes to C, that won't work. So you have to name it R2. You have to give things unique names. Or you can not name them. And remember, you're going to have to give values to Ka, Kb, Kc, and initial values for A, B, C, and D. And if you're doing this in Colab, it will helpfully suggest the syntax for you, but you better be a little bit careful because it may not actually give you the thing you want. At least when I did it, it helpfully suggested not what I needed. Almost what I needed, but not quite. And if you've got it and you want to play, you can try changing the values of Ka, Kb, and Kc, or change the initial values of A, B, and C. You get something that looks like that? So you see, you start out with A only. A decays exponentially in time. Then we get a peak of B. Then we get a peak of C, and eventually all of our A, B, and C are turned into D. Now, if we change the initial amount of A, what happens? If I make A equals 50 instead of 10, what happens? Does the amount of time the reaction takes change? Because that's the kind of question we can ask. If we change the amount of A initially, does the reaction take longer to finish? That's a reasonable kind of question to ask. What happened when you changed A to 50 instead of 10? Another one that's pretty interesting, maybe we'll do this at home, is if instead of making Ka, Kb, and Kc equal to 1, 1, and 1, you make them exponentially or related to each other. So you make them 1, 10, and 100, or 100, 10, 1, or 2, 4, 8. Try that. That's actually a little bit more interesting if my cascade is longer. In other words, if I have A goes to B, goes to C, goes to D, goes to E, goes to F, goes to G, goes to H, then having those harmonic sequences of the Ks becomes quite interesting. In one case, you can have all of the reactions happen essentially at the same time with different amplitudes. And the other case, you could have all of the reactions have the same amplitude but happen at different times. And then the last thing that I wanted people to try today was what we did here was we had a complete reaction where we started with only A and we wound up with only D. We can also have a cyclical reaction where D goes back to being A again. And that's called, that gives us a non-trivial chemical equilibrium. And so I'd like people to just, as the last exercise in our last 7 or 8 minutes, add one line to your simulation. Actually, I have to add 2 lines. Another reaction, D goes back to A at a rate KD times D. So you'll have to add a new reaction term, and you'll have to add the value of KD. And ask the question, how is that different from what you had before? Does the time series of the chemical reactions look the same to you, or is it different in some fundamental way? And if you've got the basic thing done, you can try changing the initial amounts of A, and you could also change what happens to the constants Ka, Kb, Kc, and Kd. Now, in these very simple sorts of reactions, these run very quickly, they're simple to do, but this is the kind of thing where you could also begin to think about using a neural network to surrogate. This is a little bit using an elephant gun to shoot a gnat, but one can do it. You can train a neural network to say, type in the values of A, B, C, D, Ka, Kb, Kc, Kd, and give me the time series. So that's something we'll learn how to do later in the semester. I mean, in this case, it's much, much simpler and faster to run the simulation directly than it would be to train a neural network to run the simulation. However, it's interesting to learn how to do that. And in cases of a much more complex model, like the one I was showing of the eye, in those situations, you might actually really want to be able to do the surrogate. So here's the code for doing that. And I'm probably going to have to rerun that installer. This is the old syntax for the installation, the one that the ChatGPT suggested the alternative to that you were using earlier. This OS kill is the same thing as the other one. That's that one that you had. Does the same thing. It still takes about 60 seconds to load. Okay. I'll do our import. Okay, and now we'll run it. And you'll notice instead of having all of our A turn into D, we now get equal amounts of A, B, C, and D. And now if I change the values of K, I'm going to change the equilibrium amounts of A, B, C, and D relative to each other. And so in the case of the first one, where we have all of our A going into D, the K's primarily change the timing over which things happen. In this case, the K's primarily change the final values that I wind up with. So that really was everything that I wanted to talk about for today. I wanted people to have a chance of doing a little bit of coding. And I realized that again, I don't know what people's backgrounds are, but generally people in ISC have done a lot of coding. And so I realize that this may seem pretty basic, but I promise you there's a lot to do. I'll see you next week. One of the things, again, this is a project-based class, and so next week I will hand out some possible ideas about things that could be projects, and we can start talking together about what a reasonable project might be. And again, it's going to depend a little bit on what your backgrounds and interests are. The goal is that eventually you'll have some Colab notebook that you can put out there and say, that'll look more like the ones that I was showing at the beginning, where it's basically, it's a project report where you have an explanation of what you're trying to do, some images, and then people can run that and see how it works. And ideally, you put in some sliders so people can say, when I change the rate of infection of my influenza virus, this is what happens. If I change the rate of vaccination, this is what happens. I'm thinking of that because for many years after 2020, we taught this course around viral infection modeling and immune response modeling. And then people got tired of that, so we went back to the older structure of the course. But people have done things like model P53, the apoptosis regulation in cells. People have modeled cancer therapies. People certainly have modeled the viral infection and immune response. So there are lots of different possible topics. People have modeled dosimetry and those PBPK models of drugs getting in and out of the body. People have modeled simple biochemical reactors and how they work. All of those can be written in the form of these rate equations that we do here. The other thing, as I mentioned, is that we can do stochastic models. And it's sometimes quite interesting to see how the model where you model the chemical reactions as stochastic, temporally independent events. Usually, if you have a lot of molecules, the stochastic simulation should converge to the continuum simulation, but there are times when it doesn't. And that's interesting in its own right. We've also sometimes had people model neural networks of various kinds. You can model neural network models can be not I should be careful. When I'm talking about neural networks models, I'm talking about what that used to mean, which is models of how neurons behave, rather than the kind of thing that we now, when we say just AI, ML type neural networks. But people have done those kinds of models as well. There are a lot of possibilities. One thing that that I often suggest that people do, not required, but it's a possibility, is we can find a paper, an article, a published article that's interesting to you that doesn't use this methodology, and then re-implement their work using this kind of framework. And that's a pretty good learning experience to do. It depends a little bit, again, if you're doing research where this is relevant, then by all means do what you're personally interested in. If you want it primarily as a learning experience, then finding a paper that's challenging enough to not be trivial to re-implement, but not so hard that you get stuck. I've had once or twice, I've had people say, I want to model all of metabolism with 10,000 chemical species. And I wouldn't recommend doing that in a semester project. Actually, these days with machine learning, you probably could do it in the semester project, but I'm not sure you'd learn a lot from it because you'd really be dependent on the computer to generate it. Again, my goal today was to make sure that everybody was able to do everything. I realize that maybe for some people that could be a little slow. For other people, I hope it was okay. And I try to, because the participation in this class is so different year to year, I do respond. If you tell me speed up, slow down, cover this, don't cover this, I do listen. I really do try, especially in a small class. And the goal is really to try to make things fun too. Believe it or not, there are people whose whole careers are based on this kind of modeling. both in science and in industry. One of the things that I really like about it is that the barrier to entry is low. You don't have to learn a lot of syntax. You don't have to learn a lot of math. You don't have to learn a lot of biology. And you can get going pretty quickly.
Perfect. Yeah. So when you have a big model that takes a long time to run, and you need it to run in a fraction of a second. For example, if you are deploying your model for controlling a system, a real-time system, for example, a machine or something, and your model is very complex and it takes like hours to run, which will not be the case for this class, but this is definitely a problem that people face in like engineering situations. So what you do instead is you run your simulation a bunch, generate a lot of data, and then use the data to train a neural network, and then use the neural network instead of the model to control the system.
Yeah, another that's have people heard the term digital twin? Okay, so a big area of research these days is trying to build medical digital twins. Medical digital twin has to run in real time. And so if you have a very complex model, say of human physiology, you need that to run in real time. And so solving all of these equations simultaneously may be too slow. Another thing is that if you want to apply a digital twin, you're often applying it to try to say, should I change the medication I'm giving to the patient? Well, that means I have to evaluate lots and lots of possible options. I don't run the simulation once forward. I'm basically doing an inverse problem, where I'm asking what are the treatments that I would have to give to get the outcome that I want. In that case, I might have to run millions of different scenarios for different treatment strategies to pick the best one. And in that case, trying to running forward, the time to run forward is critical. Another place that the machine learning can be used is in what's called parameter identification, which is in this case, these are completely abstract models. But suppose I have a complex model that I think represents the underlying reality, and I have some experimental time series. And I now want to do parameter fitting to get what are the values of K, A, KB, KC, and KD that I would have to get the observed thing. That kind of parameter fitting, classical parameter fitting, can be very slow. And we'll do that classical parameter fitting. SciPy has parameter fitting algorithms, and you'll discover it, at least as far as running them in class goes. You type your simulation in, you say, use SciPy optimize such and such, and you sit and you go, wait. That's another place where machine learning sometimes can give you much faster results. Now, as usual with machine learning, sometimes it'll fail on you. It'll give you no output. So parameter optimization and identification is another place where machine learning techniques can really be helpful. So in accelerating simulations, in determining interventions, optimal interventions, and in parameter identification, all of these things are places where current machine learning techniques are really that's sort of cutting edge technology. In this semester, we can only go so far in a few weeks. But I hope we can introduce you to some of these ideas that'll give you a framework that you can then use going forward to apply them in the places that you find them most useful. Surrogates are probably the first one to start with, but again, there are these other applications, control theory applications and parameter identification applications too. I've been involved quite a bit in my own work in this area of medical digital twins. And so for me personally, it would be fun to teach the course around those ideas too. So I hope that'll be fun for people. And the goal is not to make this a very, I mean, sometimes these AI machine learning courses get to be very, very heavy. You have to do a lot of mathematics, and we'll try to keep it light. I mean, if you really want heavy mathematics, I'm trained also as a mathematician. So if you want heavy mathematics, I give it to you. But my experience at ISE is generally people prefer the coding side of things to a lot of mathematics. But I'm happy to adjust the levels of the sort of the balance of those things depending on your interests. Well, thank you very much, everyone, and we'll see you next week then.